Hello everyone, in this video I will tell you what are the different gastrointestinal parasites that infect dogs and cats. I will also show the procedure for performing a flotation technique to identify the parasites present in the fecal sample. There are various GI nematodes that infect our dogs and cats. These are the large roundworms known as the ascarids. Ascarids are very common in dogs, especially in puppies. The most common species is the Toxocara canis. The eggs of Toxocara canis measure approximately 90 by 75 micrometer and are subspherical with a thick, rough shell similar to a golf ball. Each egg contains 1 or 2 cells when freshly passed. Adult Toxocara canis are located in the small intestine of the dog, and eggs leave the host in the feces. An infective third-stage larva develops in each egg after 3 to 4 weeks in ideal environment conditions. Another species under the order Ascaridida is the Toxocara cati. The life cycle of Toxocara cati is believed to be similar to that for Toxocara canis in dogs, with a few exceptions. Adult Toxocara cati are located in the small intestines of the cat, and eggs leave the host in the feces. An infective third-stage larva develops in each egg after 2 to 4 weeks in ideal environmental conditions. Several types of hookworms can cause disease in dogs and cats. Ancylostoma caninum, Ancylostoma brasiliense, and Uncinaria stenocephala are the hookworms that infect dogs. Hookworm eggs are first passed in the feces 15 to 20 days after infection. They hatch in 1 to 3 days after deposited on warm, moist soil. Transmission may result when larvae are ingested, or in the case of Ancylostoma caninum, from the colostrum or milk of infected dogs. Infections with Ancylostoma species can also result from larval invasion through the skin. Adults of the nematode Trichuris vulpis live in the large intestine of dogs and rarely in cats. They attach themselves firmly in the intestinal wall. Eggs are passed in the feces and become infective in about 4 to 8 weeks. Under ideal conditions, whipworm eggs can remain dormant in the environment for several years, unless they become dried out. Once infective eggs are ingested, larvae develop in the small intestine and then move to the cecum and colon where the adults mature. Eggs of Trichuris vulpis measure approximately 70 to 90 by 30 to 40 micrometer, and because of the polar plug at each end, they are commonly described as lemon-shaped. The samples were collected in Sinamar 1 Poblacion, Cabacan. The dog ages from 1 to 2 years old. This dog is kept inside a cage and sometimes freed outside the house of its owner, wherein stray dogs are roaming around the street. The dog also does not have irregular deworming and was only dewormed when it was a puppy before it was given. The stray dogs that it usually plays with also did not undergo regular deworming. The collected feces was stored in a container and was collected just hours before the said laboratory, ensuring its freshness. Methods in preparing the salt solution. A 200 grams of salt was dissolved in a 500 ml of distilled water. The solution was mixed using a stirring rod. Methods in performing simple test tube flotation. 3 grams of feces were measured in the weighing scale placed in a plastic cup. A 47.5 ml of saturated saline solution was added and mixed until the feces is well dispersed. The mixture was sieved several times to remove the debris until a homogeneous mixture suspension was seen. Using a pipette, every test tube was filled until a convex meniscus at the top is created before it was covered with a cover slip. The test tube was allowed to stand for 20 minutes. After 20 minutes, the cover slip was removed with a drop of fluid adhering to it.